Okay, so heterochromatin and euchromatin, it's very much important structures you can say. See, based on the, staining, they have divided this chromatin into two types, heterochromatin and euchromatin. Now let us see what is heterochromatin and euchromatin. See, whenever the DNA condenses and when it forms the chromosome structure like this, okay, whenever it forms the chromosome structure like this, okay, like this, there are some regions here which are highly condensed like this. They're highly condensed like this. Some regions where are highly condensed and some regions they are lightly condensed. Okay, means coiling. At last it is nothing but coiling only, no? So when it is too much coiled structure is found, then it looks like this. It's too much coiled. More DNA is found there, but it is completely coiled. And some regions, even they are coiled, but not comparatively, they are not as much as coiled as this. So the one which is more coiled regions you will find many regions here like highly coiled structures like this. Now the regions which are highly coiled, actually what will happen, more amount of DNA is present there. So they take more stain and they appear what? They appear darker. So when staining is done to the chromosome, some regions appear dark. They are darkly stained, you can see. And some regions like this area and here, you can see they are lightly stained. So we will just name this one. Now it is lightly stained. Lightly stained region. So based on this, they have divided into chromatin as euchromatin and heterochromatin. The chromatin region which actually stains darker, that region we call it as heterochromatin. And the region which is lightly stained, we call it as euchromatin. Okay. Now what is the speciality of this heterochromatin and euchromatin? Yes, it is very much important to understand this also. See, heterochromatin, how will you recognize it? Because it is darkly stained, we can say it is heterochromatin. This chromatin is lightly stained, we can say it as euchromatin. Now, the speciality of these two things is that in heterochromatin, again we have two types, again like facultative heterochromatin and constitutive heterochromatin. Anyways, we need to just understand what is heterochromatin first. See, try to understand here. Heterochromatin region is condensed, highly condensed form. Okay, you will not be able to get DNA freely here in this region. It is tightly coiled. And here you can get some DNA you can get easily. It is loosely condensed. Now you know that for transcription, what will happen? RNA polymerase or DNA polymerase, no, enzymes. Basically, what will happen if this is the DNA? Okay, for example, anyways, if this is the DNA sequence, what will happen? The enzymes will have to, move on this DNA, right? They need to move on the strands. They need to open up the DNA, and then they need to move on the strands. Then only RNA will be produced. Now, for the movement of this enzymes and for the attachment of the enzymes, what is required is DNA. And what has happened here is that DNA is there, but there is no space for enzymes to, exactly find out the sequences, because it's completely compact there. But here if you see, there are regions of DNA, there are strands which easily, enzymes can occur. Enzymes can do the process of transcription or any other function. So this region which is lightly stained, we call it as euchromatin. And speciality about euchromatin is that this euchromatin is transcriptionally active DNA, or Yeah, you can say active region. And DNA also is right. Anyways, it is transcriptionally active region in a chromosome. And this region, heterochromatin, because DNA is not available easily for the enzymes to function, so we call this as transcriptionally inactive region. So these are important things. Which is darkly stained? Heterochromatin. Which is lightly stained? Euchromatin. Where you will find all this darkly stained, lightly stained region? In the chromosome, and that too in the metaphase region. Metaphase stage. And then what is the speciality of euchromatin? Since it is loosely coiled, there is no much condensation here. Transcription can take place easily here. But here it is completely coiled and there is no available of DNA properly, easily. So it is transcriptionally inactive region. Okay. And I told you in heterochromatin also, again there are two types. So some heterochromatin, and one more thing. The condensation and decondensation I told you. Anytime it can condense, anytime it can decondense. That is all about the proteins. Because proteins are coiling them, no? If proteins are removed, DNA will be available there. So some of the heterochromatin, they change themselves into euchromatin. Some heterochromatin remain as permanent heterochromatin. So based on that, again heterochromatin is of two types. But anyways, that is not important now. So what is important is about to remember these two things. How to remember? Where we have studied. Bacteria, true bacteria. Means true. So euchromatin means true chromatin. True chromatin because this is acting as real chromatin, real DNA, which helps in transcription. And transcription हो गया तो क्या हो जाता है? Protein बन जाता है. So euchromatin helps in protein synthesis. They help in transcription, so they are very much important. I hope this much is clear. Heterochromatin and euchromatin region. Very, very important. And here only NEET questions. Usually they will be asking about heterochromatin and euchromatin region. Okay. Now, coming to the final part of chromosome. The final part of chromosome, we will be studying. We have done structure of chromosome in detail. But now we will see types of chromosome. We will be seeing types of chromosome. Okay, now based on number of chromosomes, based on number of centromere, based on position of centromere, chromosomes are divided into different types. Now what we will be seeing is based on position of centromere. Based on position of centromere. What is centromere? Centromere is also called as primary constriction. Centromere is a region where two chromatids get attached. Centromere is a region where kinetochore gets attached. So centromere is a region which actually separates during early anaphase. Okay. Now, so based on the position of centromere, I told you centromere may not be always at the center. It can be at any region on the chromosome. So based on that only we have divided into different types. Let us see that. Now for example, let us consider this as a chromosome. Okay, here this is the centromere region. And what are these? Now you should just tell me, fatafat. These are what? These are chromatids. Chromatid. Okay. Now this is the centromere region. What you can see here is that my diagram is not that. Okay. Okay. What you can see here is that this centromere, where it is exactly located now. When you consider this as a chromosome, where the centromere is located, it is in the exact center. It is dividing the chromosome into two equal parts. You can call it as P, you can call it as Q. P arm, Q arm. Both the arms are of same what? Same length here. So this is present in the center. Then we call it as metacentric chromosome. Metacentric chromosome. Metacentric means what? Centromere at the center, in the middle region. Then next one, if you see the chromosome, centromere, it is slightly upper compared to the center part. Then what will happen? It is slightly upper, in the upper region. Then it will divide what? It will divide the chromatid like this. One arm will be slightly longer compared to the other arm. Here you can see P is smaller, Q is longer. And then this type of chromosome, we call it as submetacentric. Submetacentric chromosome. Where is the centromere here? Little bit away from the center. Now it is dividing the chromatid into two unequal parts, right? Then next one. Now the centromere is present somewhere, okay, in this region, wherein you can see that it is almost towards the end, like this. What you can see here? I don't have space properly, so I think you can understand here. Here two arms are equal in size. Here one is small, one is slightly longer compared to this. Here if you see, it has almost towards the end, it is going towards the end, and you can see that P is very small and Q is large, long here. And this is called acrocentric chromosome. Acrocentric chromosome. Acrocentric chromosome. Here in acrocentric, centromere, where it is present? Almost towards the end. It's not completely in the end, but almost towards the end. Then finally, the last one. In some of the organisms, you will find this type of structure also, wherein centromere is on the top, and you can see that the you will not be able to see that P arm here. Only you will be able to see this Q. The centromere is completely towards the end. Which end? The end of the chromosome is called telomere, no? The centromere is found in the telomere region here. Now that is why we call it as telomeric chromosome. Telocentric. Telocentric chromosome. In telocentric, you will find the centromere in the telomere region, at the end region. There is no, you cannot see the P arm only. Completely only you can see the Q arm. I'll write that also if you want. This is called P, this is called Q. Simply just to differentiate, we have named it. That's not mandatory. Anyways, I hope you understood. So this is based on what? Based on the position of centromere. We have divided this. Again, based on number of centromeres, again we have different types. But this is given in NCERT and you need to remember this first. So there are 4 types. Metacentric, centromere at the center. Submetacentric, little bit away from the center. Acrocentric, almost towards the end. Telocentric, end region. Centromere at the end region. Now there are some other names also given to it based on, same thing only, but names, sometimes they'll ask based on the shape what you can see here. Based on that, these chromosomes ultimately after metaphase region, what they do is during early anaphase, you can see single chromatid, right? So when they are pulled away by the spindle fibers, the centromere is pulled. Centromere region will be having kinetochore. So centromere will be pulled that time what will happen? That arms will be like what? Floating, or you can say flying, not flying. Yeah, it will be freely moving. So how it looks during that time? Based on that, again they have given names for this. So please take down this much fast. Okay, we'll see that. I'll just rub this. I'll just show it here only so that easily you can get connected here. So what you can see here is that centromere will be connected. I'll show you with the help of a cell. Now let us consider this is the cell where it is moving. So during early anaphase, spindle fibers will take what? Spindle fiber will catch hold of this. I'm not showing many here. I will show only one. Only one chromatid here. So when this chromatid is moving, that time centromere will be pulled. It is pulled towards the end. What you will see, the arms. The arms will show. Arms will be like this. Remember, always centromere will be pulled and the arms will be towards the equator only. Because like this it is pulled towards that side. The other one will be pulled like this. There is other chromatid also along with this. So that chromatid will be moving this side like this. Like this. Right? So which alphabet can you, after seeing this, which alphabet you are remembering? A, B, C, D alphabet. If you see this, it looks like V shape. So this chromosome, metacentric, is also known as V-shaped structure, V-shaped chromosome. Shape of the chromosome. If it is V, in exam they'll ask you, V-shaped chromosomes are also called as metacentric chromosome. Metacentric during early anaphase or late anaphase, they show this structure which is V-type shape. Okay. Then, now you only will tell me about this also. Shall I rub this? What? Again. Okay. So here, if you see this, how it will look. I'll just show you that chromosome here then. Okay, we will see only one side if it is moving, how it will look. Now let us consider here, if it is moving towards this end, this chromatid will have what? One is shorter arm, like this. And the other one is what? The other one is longer arm. It's pulled like this, so the arms are towards the equator. How it is looking for you? Huh? You don't tell me it looks like V only. It is shorter one and this is longer one. It looks like what? It looks like L, no? So we call it as L-shaped chromatid or chromosome. L-shaped structure. During anaphase it looks like this. Then coming to this one. Here, if you see centromere, and what centromere has, very small structure, very small, because almost towards the end, and very long structure like this. Okay, here what do we call it as? See, if you show the other side, okay, if you show the other side, it looks like J. If you show the other side, are you able to understand? No, it's somewhat. Now the other side, if I show you the other chromatid, usually even if you consider this also, this type, if it is seen, it is J only, no, it looks like J. I'm not sure, like, is it looking like that or no, but remember that this is acrocentric, and what shape it has, it looks like J. So we also call it as J-shaped chromosome. Coming to this, there is no need to show it also. It looks like what? It looks like this, no? Like this. Anyways, when it is pulled, you will be able to see that it looks like this. Especially it is not found in human beings. So this is called what? This is called I-shaped chromosome, small I. So this is about types of chromosome based on the position of centromere. I hope I made it clear. Okay, and please, before understanding this one, you have to go for the previous video. Please check out that, understand that, then only come to this video, or else you'll not understand anything, because we have discussed about chromatin, chromatids already earlier, only in the earlier class. So for this, if you for this video, without preparation of that lecture, if you have come directly to, you have jumped it directly here, then you will not understand anything. And also these things you need to repeat. You people know very well, in class we used to revise again and again. Now we are not getting chance to revise, and usually if you don't revise, our brain doesn't have that capacity to remember easily for a long time. And now you are yourself, you need to prepare. So we are just going to deliver the things like this, whatever we know on the boards here, and you need to understand that. And what you need to do is, you need to practice it continuously, revise it for many times. Okay, any doubts you have, please ask, we will be explaining again. But please don't just waste your time in this lockdown. You have to be serious. It's a great time you are getting for most of the preparation, and hard work you can do now. Okay, now you can do hard work. At the end we can come for smart work, but now you have a lot of time for hard work. So please do it. Anyways, I don't want to waste your time now. Already it is time up. And I think you have understood the chapter. I mean, still so many things are there. We have not yet completed the chapter. Hope so we'll complete soon. Major things are completed. What is remaining is very small organelles. Organelles are almost completed. Small things are remaining now. So we will complete this chapter without fail. And please note down all these things and prepare well. Okay. See you in the next class. Thank you.